Experiment 6 will deal with a technique that is used extensively in the chemistry laboratory, that is spectroscopy. The concepts that we'll tie in with the lecture are the properties of light and also atomic emission. The objectives of this experiment are to look at the two main types of spectroscopy, namely emission and absorbance spectroscopy. Emission spectroscopy will be studied by looking at the emission spectrum of a fluorescent light fixture in the lab room. We'll investigate absorbance spectroscopy in several ways. First, we'll use a spectrometer to generate an absorbance spectrum, which is a plot of absorbance versus wavelength, for a colored sample solution, in this case, red food dye, and then we'll utilize the data to demonstrate how the absorbance spectrum is obtained. Next, we will prepare a series of diluted solutions of the food dye, measure the absorbance of each solution at a selected wavelength, and make a plot of absorbance versus concentration. This is known as a Beer's law plot, for reasons that will be made obvious later, but is also referred to generally as a calibration curve. Finally, we will utilize the Beer's law plot to determine the amount of red food dye in a sports drink. This represents one of the most useful applications of absorbance spectroscopy, determining the concentration of an absorbing species in an unknown solution. Spectroscopy is simply a measurement of light intensity. In emission spectroscopy, the light being measured is emitted from a source, such as a fluorescent light. In absorbance spectroscopy, light from a light source passes through a sample where some of it may be absorbed, and the intensity is measured on the other side. The device used to measure the light intensity is called a spectrometer, or sometimes spectrophotometer. When light passes into the spectrometer, it is separated into individual wavelengths of light, and the intensity of each of these packets of light is measured. Come back here. Here's a schematic of a spectrometer. The light is emitted into the spectrometer and is directed by mirrors onto the diffraction grating, where it reflects off the diffraction grating at various angles depending on the wavelength of light. The light then hits a different cell in the diode array detector where the light intensity is measured. This plot will be provided you on the post-lab sheet, and this represents the emission spectrum for fluorescent light. It's a plot of light intensity versus wavelength. You will notice that when the light is diffracted, it appears only as a series of peaks, each occurring over a narrow wavelength range in the visible region. Since the color of light emitted in the visible region depends on the wavelength, each peak represents a different color of light being emitted. We can also view the color of light emitted by the fluorescent light by using a simple handheld spectroscope. Like the spectrometer, the spectroscope also contains a diffraction grating that separates the light according to its wavelength. When we look through the spectroscope, we see the lines of the different colors corresponding to the peaks of the emission spectrum. An absorbance experiment actually consists of two separate measurements. The first is to measure the light intensity as it comes directly from the light source. In order to subtract effects due to the solvent our sample is dissolved in, and also the cuvette, which is the container that the sample is held in, we place a cuvette filled with water in the sample holder and measure the light intensity at many wavelengths. This cuvette is called the reference or the blank, and the intensities are labeled I sub 0. The second measurement occurs after the cuvette containing the colored sample solution is placed into the sample holder. Because the sample is colored, it absorbs light at certain wavelengths, and the light intensity at these wavelengths will be lower. These intensities are simply called I. What we're interested in is the amount of light actually absorbed by the sample. This can be calculated by taking the log of the ratios of the intensities of the reference and the sample. The spectrometer does this calculation at each wavelength and makes a plot of absorbance versus wavelength, which is our absorbance spectrum. When you open the report sheet for this experiment, you will see a plot of intensity measurements for the reference and for the sample that the spectrometer makes, just like this, and also the absorbance spectrum that the spectrometer calculates. What you will do is pick two wavelengths and measure the reference intensity, the sample intensity, and absorbance at that same wavelength. You will then calculate the absorbance from the intensities and compare it to the measured absorbance at two different wavelengths. There we go. In the next part of the experiment, a series of dilutions of the stock solution will be made, and also a diluted solution of the sports drink. The sports drink has to be diluted so that its absorbance will not be too high. The absorbance spectra of each solution will be obtained and plotted together on an absorbance series plot, which will be given to you on the post-lab slides. A wavelength will be selected and the absorbance value for each solution will be determined. We'll use the absorbance values for the stock solution and each of the 4 diluted solutions created from the stock solution to prepare a plot of absorbance versus concentration. You will need to calculate the concentration yourself using everyone's favorite dilution equation. This is known as the Beer's law plot. You will then use the absorbance of the sports drink and the Beer's law plot to determine the concentration of food dye in the sports drink. Now it's time to go watch our award-winning experiment video. Once you completed the video, you can go to the Experiment 6 post-lab presentation and get information on how to complete the lab report.